Hello everyone, my name is Inês Preguiça and I'm a PhD student at ICBR from the University of Coimbra in Portugal. And I'm here to tell you all about the HaPILLness project, which gave origin to my PhD work plan. HaPILLness is an initiative to move forward the refinement of oral drug dosing in experimental rodents and was recently awarded the EPAA Refinement Prize. So I'm looking forward to answer all your questions. When the HaPILLness project received the EPAA Refinement Prize, it was the highest form of European recognition we have received so far. It is truly a testament of the project's potential to become a standardized practice in European research institutes. Well, according to the last report from the European Commission, 6.8 million of experimental rodents are used each year in the European Union. Oral drug dosing is a technique broadly used in animal studies, and actually, according to the ETOX database, 77% of these studies employ the oral route. So, in sum, the welfare of millions of experimental rodents could be improved each year in the European Union alone. The main outcome is the improvement of animal welfare in experimental rodents, as they will no longer experience the stress of the invasive method of garbage. Since stress negatively influences several physiological systems, its reduction allows for an equally important outcome, which is data homogeneity, ultimately leading to a reduction of the number of animals used in a given experiment. So, with pill tech, we can achieve both refinement and reduction, and we really want you to know that animal experimentation can be performed in a friendly and reliable way. The next steps of my PhD project are to validate pill tech in animal models of disease, which mirror human diseases. But the HaPILLness project goes way beyond my PhD work plan, and we are working hard now to gather partners across Europe and get funded to perform a European multi-centric validation study to standardize and scale up HaPILLness implementation. First, I need to openly state that we researchers do use alternatives whenever possible, meaning that we use them when our questions can be answered by the simulation of a single physiological system. This is mandatory under European legislation. Yet, it is important for society to understand that before any human trial, drugs need to be screened for safety, efficacy, side effects, dose range finding in a whole organism, as multiple and interconnected systems actually impact drug response. So, if we were to skip this step, human trials would pose life-threatening risks, a consequence that we should all consider when thinking about ending animal testing.